Hello, micro students. In Chapter 10, we're going to be doing the biochemistry of the genome, talking about the structure and function of both RNA and DNA. The genome is all of the nucleic acid, and in particular the DNA that is in a cell. So we mentioned nucleic acid as a macromolecule in a previous chapter. And now we're going to look at the details of the monomers used to build nucleic acids and the reactions that we use to make that happen. We see eukaryotic organisms on the left, 2 human beings, and a prokaryotic one on the right, which is E. coli. They're used here as examples of genomes. On the left, we have identical twins. Unless you have an identical twin, there is no other human being on the planet that has the same genetic sequence as you. Their genome will be different. Identical twins began as a single fertilized egg that during early development split into 2 clusters of cells, and then those each developed into entire human beings. So they started out as 1 fertilized egg, so they are genetically identical. Fraternal twins would be 2 separate eggs that are fertilized, and then they're born at the same time but have different genetics. E. coli is put up here because all prokaryotes can only divide by binary fission. There is no sexual reproduction, so barring mutation, the offspring of prokaryotes are always genetically identical to their parents. We knew the chemical composition of DNA long before scientists knew that DNA was the genetic material. So some early experiments. This is looking at a single-celled algae, 1 alga, 2 algae, 2 to 6 centimeters in length, and we can actually see that without a microscope. So the nucleus is here in the foot structure. There is a stalk and a cap. And when the cap is removed, this organism can regenerate its cap. But if the foot is removed, then no new foot is generated. And so the information for regeneration must be down here in this nucleus. Looking at 2 morphologically distinct species, we know that cellular morphology is the shape of a single cell. So we're looking at the morphology of the entire being here. And there was the green ones with a flat cap and the blue ones with like a floral shaped cap. And if we take off the cap of a green algae and graft on just the stalk of the blue algae, a new stalk gets regenerated, but it's green, and that is because the nucleus, the blueprint, is down here in the foot. So it contains the genetic information to rebuild a green cap. So even though there's a stalk from a blue algae, a green cap gets regenerated. And if we have a blue algae, we cut off its cap and give it a green stem. Its genetic information is to recreate the blue head, and indeed it would create the blue head. And so this was some information and evidence that the nucleus contains the genetic or inheritable material. And before this point, it wasn't known that the nucleus is where the genetic material was. Even if they knew DNA is in the nucleus, it was not yet determined that DNA is the genetic material. Looking at it in fungi, beetle and tatum, looking at irradiated, so the DNA is damaged, and non-irradiated mold spores. If you allow those to mate, you're going to get some hybrid spores. And they will probably contain mutated DNA because one of the partners had been exposed to X-rays to damage that DNA. and then grow these mutants on complete media. And that means it has all the amino acids there, so it would provide any food that they would need, but also grow the same mutants on a tube of minimal media. And you'd have various types of minimal media, each of which is lacking a particular amino acid, or each of which has a single amino acid added. So complete media has all the amino acids. Minimal media doesn't have any, but they went through all 20 amino acids, so minimal media with alanine, minimal media with threonine, etc. Only one amino acid in each. And the spores that grow in only one of those tubes have a pathway that produces that particular amino acid. So again, looking at finding where there's a mutation, if they are able to grow in the presence of that one amino acid, we know they've got the enzyme for that one amino acid. This series of experiments led to the concept of one gene, one enzyme. Each of our genes is a sequence of DNA. That DNA tells the cell how to create a particular protein product. And in our cells, there are proteins that do various functions. But in metabolic pathways, the enzyme is the key protein. And so when they looked at the wild type, if they grow this bread mold that is the wild type, the normal, non-mutated strain, it will grow even in minimal media because it contains the enzymes to be able to make its own amino acids. And so even if you supplement the media with various amino acids, it will still grow. It won't need the amino acids that are provided in the media, but it will still grow. A particular mutant 1 in minimal media can't grow. But if you supplement it with the amino acids, then it is able to grow. So mutation 1 had a mutation in the first gene, but if you supplement it, then it is able to grow in the presence when it has been provided with the other amino acids. Mutant 2 can't grow on minimal media, and it can't grow if you provide it with ornithine. And that means it doesn't have the enzyme to be able to convert the ornithine to the next step. We're trying to complete this entire metabolic pathway. And mutant 3 has a mutation in enzyme 3, and so, of course, it will not grow in minimal media, but if it has ornithine or the citrulline, it still can't grow because it can't convert the citrulline into the arginine. And if we provide it with the arginine, then it is capable of growth. So by looking at individual mutants and which amino acid they required, they could determine which gene was missing. They might not have the DNA sequence, but they know that that organism is missing the genetic information to produce the enzyme, and where in the pathway is that enzyme. While this image does show mice that are decidedly eukaryotic, the experiment was looking at prokaryotic genetics, in particular the genetics of Streptococcus pneumoniae. There it is. Dr. Griffith noticed that when streaked on agar, the colony morphology, the appearance of the growth, the macroscopic growth on an agar plate, was different between certain types of the Streptococcus pneumoniae that did cause pneumonia and death versus other strains of Streptococcus pneumoniae that did not cause illness and death. It was later proved they really are the exact same genetic bacteria except for this one feature. And so the two different types of bacteria were rough, meaning when looking at an agar dish, the bacterial colonies, the clumps of growth, were rough, versus smooth. The appearance of those colonies on agars were shiny and glistening. So if you have a rough strain, which is non-virulent, meaning unable to cause disease, inject the poor little mouse. He lives. She's fine. If you take the smooth bacteria and you kill them. Heat them up so that the bacteria are dead. But any chemicals present in those bacteria will still be in that test tube. Inject the dead smooth bacteria and the mouse lives, which you might expect. The bacteria are dead, and so the dead organism doesn't cause illness. And the third one, the rough bacteria that are alive and the smooth bacteria that are dead, mix them up in a test tube and let them set for several hours. Inject that combination into a mouse, and the poor little mouse dies. When the blood was extracted from this poor little dead mouse and streaked onto an agar surface, smooth bacteria were recovered. And in the last experiment, we look at those smooth bacteria that were recovered from this poor dead mouse and put them into a live mouse, and they killed the poor mouse. So this experiment showed that some chemical, at the time they didn't know which chemical, but some chemical in dead smooth bacteria transformed the rough bacteria into having the exact characteristics of smooth bacteria. Some chemical was capable of causing that. To determine that, actually, let's go back. They extracted the chemicals from the dead smooth bacteria, and by chemical and physical centrifuge experiments could separate out the different macromolecules. So you could have a test tube of suspended dead smooth bacteria and be able to extract the carbohydrates, the proteins, the RNA, and the DNA. And then do an experiment where the carbohydrates from smooth bacteria are added to rough bacteria, or the proteins from smooth added to rough, or the DNA from smooth bacteria added to rough, or the RNA. And the final result was that only the DNA from the smooth bacteria, when added to the rough bacteria and injected into the poor little mouse, the mouse died and smooth bacteria were recovered. So carbohydrates can't do it, proteins can't do it, RNA can't do it, but DNA is the transforming chemical that gave the rough bacteria a new trait. So this was definitely evidence that DNA is the genetic material. Avery, McLeod, and McCarty followed up, and then we can see where we have the proteins, the DNA, and the RNA. If everything is present, we add the rough cells, we get smooth cells. If there's no protein, but there's DNA and RNA, we get the smooth cells. If there's no RNA, we get the smooth cells. But if you take away the DNA and just keep protein and RNA, then you do not get the transformation. So that doubly confirms that DNA is the chemical that is required to transform the rough bacteria into smooth. We've seen these before. These are bacteriophage, viruses that infect bacteria. This is another experiment that was done to indicate that regardless of the type of pathogen or the type of organism or even the type of virus that's not an organism, that the DNA is the genetic material that can be used in cells. So we've got one that is labeled blue and one that is red. This says 32P labeled DNA. 32P is an isotope of phosphorus. It's a radioactive isotope of phosphorus. And 35S is a radioactive isotope of sulfur. The DNA is labeled with radioactive phosphorus because phosphorus is found in DNA, but it is not found in any amino acid, so it is not found in any protein. The protein coat is labeled with sulfur because sulfur is found in the amino acid cysteine, which means the sulfur is found in proteins. And nucleic acids do not contain sulfur. So these viruses were specifically labeled to have radioactive DNA, and then that could be traced to see where it went. This batch of viruses were labeled with radioactive sulfur, so the proteins could be tracked. Mix the virus with poor little bacteria. It injects its nucleic acid. They were blended and centrifuged to separate the phage from the bacteria. And the lighter phage particles float and the bacterial cells sink. And when they look at what's in the bottom where the heavier cells are, the bacteria have radioactivity. When they looked at the sulfur-labeled phage, it still injects its DNA. They allow the infection to happen, they mix it up, they blend it, and the bacteria had no radioactivity, but the broth at the top, which would have the lightweight assembled viruses, had the radioactivity. So the genetic material enters the cell, and even though there's viruses in the cell, we can find that radioactivity inside the cells that are the heavier particles that sink. When the protein is radioactive, we see no radioactivity in those heavier cells. So even though there are assembled viruses, the genetic material that entered that bacterium had no radioactivity, so that non-radioactive genetic material directed the synthesis of new genetic material for the virus and new proteins for the virus. No radioactivity, we only found radioactivity in the particles that were suspended in broth. So even in viruses, the nucleic acid is what's entering into a cell and causing changes. This timeline you do not have to remember, but if you are interested in looking at any of the other discoveries along the way, we found Mendel did a bunch of heredity experiments, Mendel's peas, from which we get the term Mendelian genetics. He didn't even know what DNA was, and yet was able to determine inheritance patterns. Meischer identified nuclein by the chemicals inside the nucleus of cells. And we progress all the way until we get to 1953 with the structure of DNA and the existence of messenger RNA and sequencing of the genome. What's missing is Rosalind Franklin, whose use of X-ray crystallography proved that DNA was a double helix. Watson and Crick got the Nobel Prize. They obtained her data without her permission, and she did not receive recognition for that Nobel Prize because she had unfortunately passed away before it was awarded, and they do not give Nobel Prizes posthumously. The monomer of nucleic acids is a large molecule, the simple form. So a single nucleotide has a sugar. In DNA, that sugar is deoxyribose. It also has a phosphate functional group, and it has this thing called the nitrogenous base. The corners that are not labeled are assumed to be carbon, and so there are nitrogens within the rings of this molecule. This is the deoxyribose by itself. When we get to RNA, the ribose sugar will have a OH hydroxyl group here. And this numbering is important for how we are going to link one nucleotide to the next to build our polymer of nucleic acids. And so this 3 with a hash mark or the 5 with a hash mark is pronounced 3' and 5', and that is the number of the carbons. So carbons 1, 2, 3, and 4 are within the ring, and carbon 5 sticks up out of the ring. And that numbering is important, like I said, for how are we going to connect these nucleotides to build DNA or RNA. And it will happen from these 5' and 3' carbons. There are 4 different nitrogenous bases in DNA, and there are also 4 in RNA. We're going to start with DNA that has the cytosine, adenine, guanine, and thymine. RNA has uracil that we will see when we get to RNA structure. You do not have to memorize the structure of these. The ones with a single ring are called pyrimidines, and the ones with double rings are called purines. When we look at the structure of how the 2 strands of DNA are held together, we're going to look at a feature called base pairing. So each of these is a base, a subunit of the nucleotide. And wherever there is a cytosine on one DNA strand, its neighbor is going to have a guanine. And wherever there is a thymine on one DNA strand, its neighbor will have an adenine. And we will see that they hydrogen bond to hold those strands together. This is showing 3 nucleotides. We have the same deoxyribose sugar for all. They're color-coded just because their base that is attached is different. The base is always at the first carbon, the 1' carbon. So we have thymine attached to the first carbon of the first nucleotide, cytosine to the first carbon of the second, guanine to the first carbon of the third. The phosphate group that is part of the nucleotide is always attached to the 5' carbon. When we finish linking our nucleotides together, the terminal or end nucleotide will have a free 3' OH or hydroxyl functional group. The 5' end always has a phosphate. The 3' end always has the hydroxyl. And if there's something attached to it, we're not at the end of that DNA molecule yet. The bond happens using dehydration synthesis between the phosphate on the 3' carbon connected to the phosphate on, or the OH on the 3' carbon connected to the phosphate on the 5' carbon. The 3' OH to the 5' phosphate. And that bond is called a phosphodiester. An ester bond means you have some rest of the molecule, an oxygen, and then the rest of the molecule. So we have the rest of the molecule, an oxygen, and a phosphate, but then we have a phosphate, an oxygen, and the rest of the molecule. That's why it is a diester. We've got oxygen surrounded by molecules on either side of it. Twice makes it a phospho because that's in there. Ester bond, phosphodiester. This would continue hundreds to thousands to billions of times depending on how long a DNA molecule is. This is just one strand. We also have to have the second strand. That will form this double helix, which I am not an X-ray crystallographer. I could not have told you that was what a double helix looks like if you freeze its crystals and bounce X-rays off of it, but that is it. So without that data obtained without Franklin's permission, Watson and Crick would not have broken the code to DNA structure, or at least not as quickly as they did. And I provided a link to an article in The Guardian about that. There is Rosalind herself. Watson and Crick were building ball and stick models of DNA. So that is an incredibly complicated process. You saw all of the atoms that went into building just one nucleotide, and they were building dozens and dozens of nucleotides and then trying to put them together into that helix form. We saw just 3 nucleotides linked for one strand a few slides ago, but DNA is double-stranded. The bases face inward toward each other, and they have hydrogen bonds between them. So what's called the phosphate sugar backbone is covalent phosphodiester bonds. So here's our phosphate to the sugar, to the phosphate to the sugar, and those are all covalent bonds. The base is attached to its sugar by a covalent bond, but we do not bond the base of this nucleotide to the same strand's base. We are binding by hydrogen bonds to the opposite side. So this is one complete DNA strand. We have a 5' phosphate, a phosphate on the 5th carbon, and a 3' OH, that hydroxyl group on the 3rd carbon. We see the hydrogen bonds between the bases that are in the interior of that helix, the spiral staircase. Anytime there's an A, it is directly across from a T. Anytime there is a C, it is directly across from a G. Adenine-thymine pairs in position only have 2 opportunities to do hydrogen bonding, but cytosine-guanine pairs have 3 places that they can do hydrogen bonding. So GC pairs are stronger than AT pairs. And when we look closely at the second strand of DNA, it is upside down relative to its neighbor. The 5' end is now on the bottom, the 3' end is on top, and even if that wasn't labeled, we do have the numbers on our carbons, and the phosphate is sticking out over here, so that has to be the 5' end. This end has nothing else joining to it, so that is the 3' OH, and our bases are stuck to the 1st carbon of each of these. And you say, why does it have to be upside down? Science isn't really good with the why. We're great with the how. The only way to get this spiral staircase that is very uniform in its width the entire way is to have one strand upside down to the other so that you can get the hydrogen bonding. If it's not, the hydrogen bonds don't work out. We also have to have a double ringed base pairing with a single ringed base. And that is what keeps the width of the whole molecule even. Why can't it be a wobbly and curvy, skinny sometimes and wide at other times? Well, it's the genetic information. For it to be uniform in width makes it much more recognizable by enzymes for doing DNA replication, making a copy of that DNA, or for transcription, where a different enzyme called RNA polymerase has to recognize DNA and read it to make RNA. If it was very irregular in shape, it'd be very difficult for enzymes to be able to figure out how to bind. And I have here at the bottom, the human genome is 3.5 billion nucleotides. That is a lot of phosphodiester bonds and an awful lot of hydrogen bonds. So there's our complementary base pairing. We have a thymine across from an adenine and a guanine across from a cytosine. 5' end on the top for this strand, 5' end on the bottom for the other. We are going to be spending another chapter looking at how does DNA actually get copied. But this image shows in our native state, we've got 2 different strands of DNA. We can denature it, make it lose its nature by exposure to high heat. That will break those hydrogen bonds between the strands. Or we can expose it to chemicals, in this case, a high pH, high level of hydroxyl ions. That will disrupt those hydrogen bonds. And when they are single-stranded, they are not stable, and so we can remove the chemical that we used to denature them, or we can lower the temperature and get the strands to stick back together. Barbara McClintock is shown here because she discovered transposons, which are also called jumping genes. Many species have the ability with enzymes to make nicks in the DNA, and that gene can lift up and move and be inserted in another region of the genome. Now we're going to look at RNA. The sugar in DNA is deoxyribose. And that is because ribose, which is the sugar in RNA, has 1 more oxygen. So if we deoxy, we take out an oxygen, we have a hydrogen at the second carbon. Ribose has an hydroxyl at the second carbon. And again, thymine was a base that was found in DNA. Thymine is not found in RNA, but uracil is. The difference between a thymine and a uracil is that thymine has a methyl group, CH3, attached here, and uracil has only a hydrogen atom. So uracil is only in RNA, thymine is only in DNA. The adenine, cytosine, and guanine are found in both, and they are chemically indistinguishable. DNA is typically double-stranded, especially in cells. Viruses are completely different. Viruses can have single-stranded DNA, but cells need that double-stranded RNA. The DNA. Oof, I read ahead. Cells must have double-stranded DNA. In cells, we have single-stranded RNA. It can fold on itself and have hydrogen bonds between complementary pairs like a guanine and a cytosine and an adenine and a uracil, but that's just localized hydrogen bonding. It's still only 1 strand of RNA. When we get to protein synthesis, we're going to be looking at 3 different kinds of RNA. Messenger RNA that contains the information that will tell the cell what order do we need to put amino acids in to build a protein. We will have tRNA, or transfer RNA, that delivers amino acids. And we will have rRNA, the ribosomal RNA. Ribosomes are made of RNA and protein, and the specific RNAs found in ribosomes is ribosomal RNA. When we get to our chapter talking about protein synthesis, we will see that the messenger RNA is linear. Our transfer RNA has several looped regions, and at the 3' end, the end with the OH, is where it will bind to an amino acid. That is not random. There is a particular code in the messenger RNA that will tell the tRNA that has the matching code, the opposite code. Because if the mRNA is GAG, the base pair for that is G would pair with C, A with U, G with C, temporary hydrogen bonds to hold that tRNA in place. And that code says this CUC tRNA needs to deliver glutamic acid. Each of the 20 amino acids has at least one of these codes that will dictate it to be bound to a tRNA. That whole thing gets delivered to the ribosome in order of the letters on the messenger RNA to build the protein. This is looking at the same culture of Serratia marcescens. So from one broth tube, streak two nutrient agar plates, but one of the plates was put at 28 degrees centigrade and the other was put at 37 degrees centigrade. The exact same tube of bacteria used on both plates. But at 28 degrees, it turns red. At 37 degrees, it does not. So even though it's genetically the same bacteria, environmental conditions can affect which genes get turned on or turned off. This would be a eukaryotic chromosome. We have to coil our chromosomes around proteins called histones, and we have both coding and non-coding regions. The genes are called coding regions because they code for a very specific product. They will be red to make the RNA, and the RNA will be red to make the protein. We have significant areas of non-coding DNA that we used to think was junk DNA, that it didn't do anything. We're now discovering that those pieces of non-coding DNA are pretty critical for how the DNA wraps around those histones to compact and fit inside cells. And it can also impact on which of the coding regions are showing or available in different cell types. Eukaryotes, we have our nucleus, our genome is bound within there, but that is our nuclear genome. As animals, we also have a mitochondrial genome from our endosymbiont theory. Mitochondria are an organelle. They are not an independent living thing, but they do have their own DNA, and they divide independently of the cells they reside in. So the mitochondria have a genome. In cells that photosynthesize, in eukaryotic cells that photosynthesize, they have chloroplasts that also have a genome. So as animals, we have a nuclear genome and a mitochondrial genome. Photosynthetic eukaryotes have a nuclear genome, a mitochondrial genome, and a chloroplast genome. And the picture on the right we saw before, when epithelial cells are infected with human papillomavirus, it damages the nuclear genome and can cause unregulated cell division. In prokaryotes, their chromosome is a single circular piece of DNA, and this is Bacillus cereus. Bacillus anthracis has the chromosome, but it has two other pieces of DNA called plasmids. One plasmid encodes a specific toxin, and the second one encodes a different toxin. Bacillus anthracis causes anthrax. And these plasmids, because they make toxins, they encode the genetic information for making toxins. Those toxins are called virulence factors, the capacity to cause severe illness. Genome sizes can vary dramatically, and that dramatic variation is somewhat related to complexity, but not entirely. Here we are with our 3 billion nucleotides. There are plants that have at least that many to even more. Other animals vary also dramatically. Yeast, single-celled fungus, coming in equal to the low end of plants and animals. A typical prokaryotic cell, a bacterial cell, Much smaller, we have only 4.5 to 5.5 million base pairs, so that's 1,000-fold smaller. And viruses are smaller than that. The largest virus, Pandora virus, 2.5 times 10 to the 6, so 2.5 million nucleotides, down to the smallest virus that only has 1,700 nucleotides. So genome sizes, tremendous variation. This final slide is an example of one sort of question I might ask on an exam. If you were given the percentage of just one base, you can determine the percentages of all the other bases. Because if you know the percentage of thymine is 22%, then, let's see if I can have any shot of writing this in. Let's do it in blue. We know that thymine is always located directly across from adenine. So they have to have the same percentage because each thymine needs its own adenine. So that would also be 22%. All right, well then how do we know how much guanine and cytosine there are? Well, if we have adenine equals 22%, and thymine is also 22%. Then we know the total of adenine and thymine is 44%. Well, that takes care of that. But if we want to get to the guanine and cytosine, then we can do, it has to add up to 100% minus 44%. And that is 56%. Well, that's all well and good. 56% has to be the G plus C. So if we divide 56 by 2, we find out how much goes to guanine and how much goes to cytosine. That's a 2, trust me. Or not. Can we erase that? Yes. Just that. I don't want to erase all of my ink. Alright. Divided by 2 equals 28%. 28, yes, 28%. So given the percentage of any one nucleotide, you can determine the percentage for the other 3. That is our overview of the structure of DNA and RNA. We will get into the function of DNA and RNA in our genetics chapters later on.